Hello, this is Jesse, and in this tutorial I'm going to go over setting up RAST, at least the RAST account for now, and then we're going to use antiSMASH to find biosynthetic gene clusters. We're going to cover codon bios for a given organism. We're also going to use NCBI's Open Reading Frame Finder, and we're going to use GeneMark for gene prediction. We're going to be using prokaryotic genes and genome, or DNA and genome, to actually we're just going to be dealing with prokaryotes for this instance. In the next gene finding tutorial, we'll be going over eukaryotes. So first, setting up RAST. So RAST is an annotation service. You submit a genome and it will annotate your genome. And annotation is basically finding all genes in the genome. So what you can do is set up your account here. I provide the link, copy and paste this to your browser, and you can set up your account here. And this is, I'm not going to show in this tutorial RAST in particular, but just want to get you to set up your account first, and then in the future tutorials we will be covering RAST, the web browser base, and then the Iris command line environment. So we will be using a RAST a good portion, or at least in a few tutorials, and you will cover RAST in the homework and your project or your presentations. So it's good to go ahead and get this set up already. Next, we're going to use antiSMASH to find biosynthetic gene clusters. So these are gene clusters, or these are genes grouped together in the genome that work together to synthesize a particular product. In this case, we're going to be looking for non-ribosomal peptide synthases. They synthesize polypeptides. So what we can do first is, as an example, we're going to work with an annotated genome sequence. So you can go to NCBI, go into taxonomy, and then here we can go to Bacillus. Bacillus subtilis. And then in this case, we just go to genome, go, and I already have all this worked through. Okay, so you just follow along. Once you get to the point where you already have the genome that you want, so once you go through and clicking on Bacillus subtilis, and you scroll down to the reference sequence session number, we're going to use this to submit to antiSMASH. Now, like I said, antiSMASH is a website that will find biosynthetic gene clusters. We go to antiSMASH and go to bacterial version, submit bacterial sequence, and then from here you're going to type in the accession number. Data input is going to be the NCBI accession number of desired sequence. So NC_00964.3. You're going to submit this, and you keep default settings for now, and then you click submit, and it's going to run. It's going to submit your job, and it's going to start running, and then you'll get results back. I already have the results already run here. So it's going to give back, I click on overview, this is what you're going to get back at first. You'll get any predicted non-ribosomal peptide synthesis or polyketide synthesis, so any type of enzymes that work together to synthesize some type of biosynthetic product. It's like antimicrobial peptides, fatty acids, and so forth. In this case, I'm just going to focus on one of them. You can look at other ones if you want, and it even tells you what they predict to be. So surfactant is a non-ribosomal peptide synthase product, we click here, and we go to, oh, actually I did not want to go here. Go back, click on overview, and we click on, notice how where it says surfactant, and you scroll right here and it says region 2 right here. Click on 1.2, and then here we are at the surfactant non-ribosomal peptide synthase, okay? And it's going to give us some of the annotations of the biosynthetic gene cluster, okay? And it color codes it based off of their annotation and what they probably do. And here's the core biosynthetic genes. We click on, so we have 3 clusters right here. So we can click on one, and then on the right it says gene details, surfactant synthase. So it synthesizes surfactant. So here it goes right here. We have in this biosynthetic gene cluster, we have anti-adenosine monophosphate binding. We have the condensation domain. So we have particular domains right here. You don't have to know exactly what they do, but just showing you what this service provides. Okay, this anti-SMASH is finding and trying to predict if there's any non-ribosomal peptide syntheses or biosynthetic gene clusters that synthesize these enzymes. And it tells you, you can click on these, and it gives you information about what domains are occurring, what features are occurring in this segment, this reading frame right here, right? So you can click on this one too, right? It gives you some information. Thioesterase, so that's that. And then there's the detailed domain annotation. So in non-ribosomal peptide synthases, they're multi-domain proteins. So here it does a nice job of annotating where this domain is at in the protein. So we have a C domain, A domain, C domain, A domain, C, and then at the very end we have these TE domains. And what happens is that you see right here, it even has a predicted product. So this protein right here is predicted to synthesize this product right here. So amino acids come through and get incorporated through this non-ribosomal peptide synthase, and at the very end you have this product. Okay, and you can also look through a database of non-ribosomal peptide synthase products. So we go to here. So this is the predicted product. If it's closed off, you don't see this. If you don't see it at first, you might have access by just clicking the plus symbol. Okay, click the plus symbol and it'll give you the picture. So if your homework asks for any predicted products, this is what you will do. Go here and you'll snip a picture of the actual end product. Sometimes if you're going through the database, or if you get the results, so we go back to overview, you might not get any particular non-ribosomal peptide synthases. So that means here you would say that there were no known predicted non-ribosomal peptide synthases using anti-SMASH. And maybe there's more than one reference genome that you can use, and you might need to use that particular genome. Or it might be, if you're giving a eukaryote, which I'll go over later, you might have different chromosomes to look at, and you can look at different chromosomes and see if there's any biosynthetic gene clusters in that chromosome, which I'll cover in the next tutorial going over eukaryotic gene finding. And we can also click on another non-ribosomal peptide synthase. Why do I keep doing that? Go back, overview, here, section, region 9, so click on 1.9. And then here we have this particular biosynthetic product from this certain non-ribosomal peptide synthase. And this one's a little bit different, right? Looks a little bit smaller than the other one. And we can also look at the product if it has any predicted product, which this process, which anti-SMASH does have. And it also gives us the annotated domain. Here we have the condensation domain, adenylation domain. We have all these domains, right? And then we can also look at this product in the database. We can click on strict. It's going to do a search for us. And here's the product predicted, and here's the similarity. We can click on the peptide, and it gives us more information about the product that is predicted to be synthesized from that gene cluster. We look at the structure. Okay, so here's the atomic structure. Look at the organism, taxonomy, and whatever you might be interested in. Okay, so all I did was do a strict search here. I'm able to look this up. This is the predicted search, and then we did a database search for any similar products. Okay, so this is anti-SMASH, and just remember, we're just looking for a particular gene. We're basically searching through the genome for a certain subset of genes. In this case, we're interested in non-ribosomal peptide synthesis and any type of biosynthetic gene cluster. And non-ribosomal peptide synthesis happens to be a subset of these biosynthetic gene clusters. There's other types of biosynthetic gene clusters that produce metabolites. So that's anti-SMASH for you. We're going to move on. So the next section is codon bias. So we're going to be looking at this codon database. And in this case, we're going over Bacillus subspecies LU11. Input it into the search box and click submit. And then we can click on the name. And it brings us the codon table for this organism. And then your homework, you're going to get asked similar questions to these right here in this tutorial. So according to the table in question 1, which codon configuration of proline has the highest bias? So all you do is find which codon corresponds to proline. So in this case, we just have this set right here, proline. So that'd be over here. And then you just look at whoever has the highest bias, so which one occurs the most often. So this is the frequency per 1,000. So here we have CCU with 19.8 times it occurs 19.8 times for every 1,000. So this would be the highest bias. Next question, according to the table from question 1, which codon configuration of arginine has the lowest bias? So same thing. You're going to look for arginine. So we have two sets, arginine here, arginine here. Then what we're going to do is we're going to look at the corresponding table over here. And here we have CGC with 0.6, really low number. And then we have arginine over here. These two. This is the lowest out of the 6. So this would be our answer right here because it has the lowest frequency or the lowest bias. Next is just looking for the codon configuration which appears the most. So in this case, you just look for the one that has the greatest frequency. So the greatest frequency is going to be the one with the greatest number. So we just search through here, and then we have AAA 63.7, and no other codon appears more than 63.7 per 1,000 observed. So this would be our answer. And then the amino acid corresponding to AAA is right here, which is lysine. Continuing on, I just answered this question, which codon configuration, which amino acid corresponds to this codon, which is lysine. And then in question 1, how many times does the stop codon appear? Well, in this case, this is simple. You just count the stop codons. You have UGA, you have UAA, and UAG. So we have 1, 2, 3. So we have 3. Then next, according to the table in question 1, what percentage of all codons are stop codons? So here we just look at the total amount, 1,617 codons. And then we have a total of 3 stop codons. So the percentage is 3 divided by the total amount of codons looked at. Divide that, 3 divided by 1,617, times that by 100, and then you get around 20%, or not 20%, 0.19%. Next, we have NCBI Open Reading Frame. So we're going to be using the Open Reading Frame Finder to find some possible proteins that are encoded in 2 sequences that are given. So we're going to go to Open Reading Frame Finder. Here it is, Open Reading Frame Finder. You can just type in, I already give you the URL right here, so you can copy and paste this and put it into your browser. So I'm going to take sequence 1, copy and paste the sequence. I'm not going to keep the parameters the same. I'm going to click Submit. Once the Open Reading Frame Finder is done finding any open reading frames, we're going to look at the one that is predicted to be the protein, right? So it's going to give back the longest amino acid sequence, which is Open Reading Frame 1. And then for us to find the possible, so some cases, not every single time, the longest reading frame is the actual protein, okay? Most of the time it's the longest amino acid, but sometimes it's not. It could be other ones. So you could always investigate every single one of them. Maybe not, you probably wouldn't want to look at too short of ones, but it's possible that this could be a protein too. But majority of the time, it's going to be the longer amino acid. Okay, so we're just going to click Blast. So now we're just going to figure out what this protein is. What's the identity of this product? Okay, once this loads, okay, let me try this again. Okay, Blast. Okay, so once we get our results back, we can click on Quick Blast. We can click on Non-Redundant and then Blast. Okay, now we have our blast results. Looks like this protein being encoded from the nucleotide sequence we looked at is drosomycin, which is an antifungal peptide. We've seen this protein before in previous tutorials. So we can put that as the answer, drosomycin from Drosophila melanogaster. So that's Open Reading Frame Finder for the first sequence and identifying the protein product. Next, what we can do is we can go back and to Open Reading Frame Finder and submit sequence 2 now. Copy and paste, submit. So we have a lot of open reading frames in this one. All of them are pretty short, except we have this one long open reading frame, and it's apparently 563 amino acids. So we can click Smart Blast, or we can click Blast. And then we can do the same search, quick blast. Now once we have our results back, we can look at the top result to help us identify the protein. So here we have the protein here. So we could snip this. And we could say that this is probably the protein being produced from this sequence that we were given, sequence 2, and snip the top result. That'd be the answer. So next, we're going to be using GeneMark for gene prediction. So we're going to find the genes in the sequence below, and we're going to snip the sequences in FASTA format. So we're going to use the GeneMark HMM tool. So here is the URL. So you can copy and paste it into your browser, and you should be brought to this page. Next, what we're going to do is we're going to take the nucleotide sequence, copy and paste. Next, what we're going to do is we're going to click gene nucleotide sequence. Then we're going to go down, make sure we click the heuristic meta-GeneMark parameters and the 2010 publication. Okay, and then we're going to submit. Okay, so now we can click on nucleotide sequence here. And then here is our results. So these are the predicted genes from this group, from this nucleotide sequence above. And you can output the sequences into your Word document. In the homework, you will get similar questions. You may be asked to actually identify the protein products from these genes. And one way to do that is to go all the way back and click protein sequence 2, and then click start GeneMark, and then you just go to protein sequences, and then here's the protein from those genes. The nucleotide sequences are translated to amino acids, and then you might be asked to identify each one. One way you can do that is you can copy and paste. You would need to remove this extra tag right here, empty FASTA definition line. Remove this if you need to, if you get any errors when you're submitting it, but you want to submit it to BLAST. You can copy and paste this whole thing and put it into your own file, and you can save it. And then if you want to blast all of them at the same time, just go to protein blast, choose file, and let's say you have a whole bunch of protein sequences in your file. Click open, and then you should click blast search, and it's going to bring you, if I do the blast search, I'm going to be able to look through each individual result. Okay, so this is a lot better than opening up all these tabs and then looking at each individual blast result on each tab. You can do it all in one go, and you can click. If I had 3 genes, if I had 3 proteins from this genes, in this case I have, I think 8, then you can just look through each of the blast results here and filter through them and click which ones you want to look at, identify the protein, and that's how you would answer those. But that's it for this tutorial. I hope this is helpful.